Welcome, everyone. Welcome to San Francisco State University's Romberg Tiburon Center for Environmental Studies. It's a real pleasure to have you all here today. My name is Karina Nielsen, and I'm the director of the center, but I just started a few months ago, so I may be a new face to some of you who have visited with us before. I'm really excited. We have full house tonight, and we have a really great speaker. I know you're going to enjoy the presentation. I'd like to take just a minute to honor our donors who have helped endow this series. So it's a recurring series that happens twice a year. We open our doors to the public and we try to bring really engaging speakers in to talk about the science of the day that affects marine and coastal environments, and hope that you will have an opportunity to engage with the speaker and ask questions and learn more about things that interest you relative to the presentation. So our honored guests tonight are Dick and Barbara Rosenberg, who have endowed the fund that underwrites this, and I would like to introduce Barbara Rosenberg to say a few words for welcome you. Thank you.
I can't tell you how honored we are to have this privilege of having sponsored this program, and I'm just thrilled and amazed and awed at the wonderful turnout on such a rainy night. Some of you know that we were turned on by visiting the lab here of an ARCS, Achievement Rewards for College Scientists, visiting her lab, and she insisted that I spend the day and Dick spent the day, and as a result, we have this program or series. And I learned tonight that Dr. Palumbi was also an ARC scholar, so the wheel goes around. So it's a privilege to be here and to be able to participate and do what we were able to do. Thank you all so much for coming. Thank you.
Thank you. I'm just going to really turn this over to one of our faculty members, and actually her name is Dr. Sarah Cohen. She actually studied with Dr. Palumbi for a while, so she knows him well and his work well and their colleagues. Dr. Sarah Cohen works on genetic issues related to marine organisms, and she's going to introduce our speaker tonight. So, Sarah.
Thank you, Karina. Before I introduce Steve, I'd like to take another minute to thank Barbara and Dick. This has been a really special semester for me because two of the most important mentors for me in my career got to come and speak here at San Francisco State at the Romberg Tiburon Center, thanks to their generosity in funding both this event this evening and our semester-long seminar series. So earlier this semester, my PhD mentor, Dr. Richard Strathman, came from the University of Washington and Friday Harbor Marine Labs. And now my kind of postdoctoral and then some advisor, Steve Palumbi, has come. So thank you so very much. I wanted to also just elaborate a little bit on what this means, not just for me personally, to have a great time yucking it up with my buddies and thanking them for mentoring me, but all my students get to meet them as well. They get to have lunch with them, hear their seminars, get career advice from them, think about next steps, and actually in the case of one of my master's students who finished last year, Steve actually welcomed her into his lab. And so Beth Sheets is down there now working with him. And because it's only fair it be an exchange program, he did send up one of his undergraduates who's now starting as a master's student in my lab, Tim Fuller. So it's great to have this partnership, and it's really great to have this kind of collegiality and mentoring going on. Thank you so much. I also have to thank Barbara, of course, because one of my students was one of the ARCS fellows who invited her to come. And so I always tell this story, write your thank you notes. Craig had lunch at the ARCS fellowship lunch, sat next to Barbara, told her all about what baby copepods eat and how she figured this out with incredible molecular trickery. And they had such a good time that she wrote Barbara a thank you note and invited her to come visit. And so the rest, I guess, is history. There were more ARCS fellowships. There's one from Dr. Stillman's lab as well that Barbara sponsored. And so it's just been really wonderful for us. And I was really delighted to see that Steve is part of this tradition as well. So thank you again. So with that said, Dr. Palumbi is really remarkable. So I first got to know him when I was a starting graduate student at the University of Washington, Friday Harbor Marine Lab. So again, it's another example of how marine labs foster this community, as we have here tonight, but that goes on all day long and all night long, where students and faculty get to meet each other. So Steve had finished, but was coming back to visit some of his colleagues, and I met him then. But when I really got to know him was at SFO, San Francisco Airport, because Steve's career in a nutshell, if you don't mind, he's a fellow Marylander like me, and he went to Johns Hopkins University, and I imagine Chesapeake Bay had something to do with his interest in the ocean, because it is an amazing bay. And I have to put in a plug for Chesapeake Bay, because when I first moved to the West Coast, everyone said, the bay, this and that, the bay. And I said, which bay? There are so many amazing bays. Anyway, after Johns Hopkins, he moved to Seattle, went to the University of Washington, where he did his PhD, as I said, working with one of the most famous marine ecologists of all time, actually, Dr. Payne, Robert Payne, who more or less invented one of the major concepts in marine ecology. So Steve took that page from Dr. Payne's book and about 1,000 other pages from everybody else's book, because in reviewing Steve's CV, which includes about 170 peer-reviewed papers, not to mention essays for the general public and several books and manuals on how to do genetics. When you look at actually just the first 10 papers on his CV, they pretty much cover the gamut of biology. I don't quite get how this was done. Maybe someone could eke this out of him in question. So having done that, he actually moved immediately to a faculty position at the University of Hawaii. And I think had a fabulous time there, starting his own genetics lab and becoming a geneticist, really. I mean, he must have done this somewhere on the side, but his thesis had actually been something completely different. Anyway, Hawaii was very nice, I think, but Harvard called. And so Steve packed up his family in February, I believe, winter, and moved the family and the lab to Harvard, to Cambridge, Massachusetts. And actually, it was before February, because I met Steve, as I said, at SFO many times as he came from Hawaii to go to meetings all over the country. And we would be waiting for our flights, and I got to know him. And I was lucky enough for him to move to Harvard, for me to need a place to do research and to be invited into his lab. So thank you very much. I'm sorry to be so long-winded about this. I will finish. From there, Steve spent a mere 6 years at Harvard before he realized that his dream was to go back to Hopkins Marine Station, where he'd spent a sabbatical. And he and his entire family had felt this was nirvana. And so, why not go back and be director? So that's pretty much what he did and where he's been ever since. And so I hope I've given you a flavor for the amazing things he does. The stuff he's going to talk to you about tonight will show you the non-academic or the mixed academic side, which is so very important to us. So thank you very much for your patience, and thank you, Steve.
So now you know that I can't keep on a topic or keep a job. So it really is a pleasure to be here. I've been up here to the center many times, many old friends here, new people who are old friends, and as Sarah has said, marine biology is really a community of people working together, more and more interested in the incredible life in the oceans, saving the oceans, giving the oceans away to the next generations. And partly that's what excites people to come and do marine science, and it's also, I think, what excites people about the sea. so that's why we've started this other thing of not just writing papers and giving science talks, but also to try to share what we know about the ocean and to share our excitement about it and how we think about it. A lot of people think they know an enormous amount about the ocean because of Shark Week. That's what you know about the ocean from Shark Week. But it turns out that sharks are not very extreme at all, except one end is pretty extreme. But they're not the fastest fish. They're the biggest fish, but whale sharks are not like that. They don't live the deepest. There's a lot of other amazing life in the oceans. And so this project, the Extreme Life of the Sea, was built around the idea that there is an incredible amount of amazing life in the ocean, and that what we really wanted to do was to show how amazing you could know about it, and that if you actually just delved in it a little bit, you would find that it was just a sense of guiltless wonder about the diversity of these critters and how they live their lives. So that's what the book is about. I needed help with this book, and so I went to the best writer that I know, who's my son. He's a novelist, and so the idea here was actually to do an experiment of kicking environmental writing into a different level. How could you take the roar of a good novel, scene building and characters and excitement, and combine that with science, which is kind of dreary, right? And build something that really brings the life of the ocean in an accurate, scientific, but exciting way. Turns out it is not hard, because the ocean is full of such amazing things. And so we had to pick and choose about what we were going to cover. And what I'm going to do in a short period of time, they've asked me to speak for only about 20 minutes so that we can have some conversations and have more of this interaction that really characterizes marine science. So I'm going to give you some stories about the life in the sea. I'm going to show you more marine species than you can possibly imagine in 47 seconds, and that'll all qualify you as marine biologists. And we can talk about the incredible diversity and wonder that's out there. I want to start with a seemingly simple thing: the fastest fish in the ocean. What is the fastest fish? And the fastest fish is like this. This is a marlin. It's a sailfish, actually. And billfish are incredibly fast. They can swim up to 40 miles an hour. They do this in order to capture food, but they don't do this in order to skewer fish on that bill. They do not use that as a skewer. What they do is to use it to bat at their food. So this is a picture from the back of a fishing boat in Costa Rica, and you can see this fish batting away at the bait fish behind. And that's what billfish do. They smack at them in order to capture them. Well, they have a problem. They're smacking the fish, and they're turning and they're gulping it down at 40 miles an hour. It's like stopping for coffee without stopping, actually. So how do you do that? The bill's 2 feet long, and by the time they smack a fish and are moving through, they have 1/30th of a second in order to actually snap that fish down. And the real challenge in building a fish that fast is how they can eat at those speeds. So what they do is they've actually built heaters into their eyeballs and their brains, because their eyeballs and brains were not working fast enough at cold ocean temperatures in order to do that. So these fish have heaters behind their eyes and heaters on their optic lobes in order to process information at those speeds. So that actually brings us to this creature. So this is one of your own. This is a marlin that we got from here. And the problem that we were having was that I really wanted to show people what it was like for a fish to swim at 40 miles an hour. And that other video was only going about 12. I searched and searched and searched. I asked people. We tried to do it. It just couldn't be done. So I called around Brooke from down here had a stuffed marlin. So we got it, and we tied it to the roof of my car, and we drove it up and down the Carmel Valley at 40 miles an hour to see what it was like for a fish to swim at that speed. So do you want to see it? Are you ready? Don't blink. That's it. You want to see it again? That's what it looks like. And that is what it looks like. If you're in the ocean and a fish is swimming by at that speed, that's how much time you've got to see it. So what we think about, the fastest fish, we think we know what that means, but actually when you're in the ocean swimming, trying to be a fish, then in fact there's all these other issues that pop up. Well, the diversity of the life in the sea is so intense that and wide that I needed in these talks a way just to get you lots of information into your brain really quickly. So what this is a 47-second rock and roll montage of about 80 different species of marine life. So I'm just going to roll it, and then you'll all get your cards for being marine biologists. There. That was a master's degree amount of marine biology in there. It was also music from my daughter's rock and roll band, Direct Divine. So she felt jealous that I wrote a book with Tony and not with her. So what other kinds of things can we sort of explore to see how marine life actually does what it does? Because it lives not only in strange ways, but in very extreme places. So let's go to the coldest parts of the ocean and see how marine organisms do that. The Antarctic is where we're going to go, and this is what it looks like. This is a fabulous picture by John Weller, who allowed us to use it in the book. This is 80 feet deep on an Antarctic shore. The water is crystal clear. You cannot see it, and what looks like clouds in the sky is actually the ice on the surface of the sea. And down all along the bottom there, these white creatures growing are not alive. These are not creatures. That's ice. Because this water is -2 degrees centigrade. It's below freezing. And that's a problem. It doesn't freeze because it's got salt in it. But it's cold enough so that ice crystals can form and grow. And sometimes they grow large enough to actually grow over those starfish and just entomb them until the summer comes. The starfish are okay because although they have fluid inside them, the fluid is the same salinity as seawater, so it doesn't freeze. But what about fish? Fish have salty blood, but it's no more salty than our blood. And then in that temperature, our blood would freeze solid. And theirs would too. So that is the problem that Antarctic fish face, that their blood would freeze solid. And now that you're all marine biologists, you know that fish with frozen blood don't do so well. And they can't live that way. So these fish, Antarctic ice fish, have a special protein that they have evolved. It's an antifreeze protein. And it's the most common protein in their blood. And it does just one thing. It binds to ice crystals that should form in the blood supply of these fish. And by binding to the ice crystals, it means that they can't grow. And if they bump into one another, they don't stick and get bigger. So the ice protein keeps the ice crystals from growing and means that these fish can live in super cold water. It's so successful at it that 95% of the fish in Antarctica are in this family that have this protein. And it allows them to live there. So we have used it too. The gene is known. We've pulled the gene out, not me personally, but geneticists have, pulled the gene out of, in fact, an Arctic version of this, the Atlantic eel pout. And it works if you allow it to be made into proteins. it works as an antifreeze. So the gene has been cloned and put into yeast, and the yeast have been used to make something called the ice-suppressing protein product. And that, in turn, has been used to solve one of the most critical and amazing problems that human society has on the planet, and that is, what about ice cream? Regular ice cream has enough fat in it so that when you put it in the freezer, ice crystals do not form, but low-fat ice cream does not. And if you are on a cholesterol diet like I am, you can only eat low-fat ice cream, and it would freeze in the freezer with ice crystals. So what happens is that a very small amount of this ice-suppressing protein product, so small that it does not have to appear on the label, is put into the ice cream in order to keep the ice crystals from growing. And that means I can enjoy low-fat ice cream without it being all ruined by being in the freezer, sort of from the fish to the technology to you, all in a couple jumps. So you will get over this, by the way, in about 6 months, and you'll go back to eating ice creams. So that's a place. Well, how about a lifestyle in the ocean? And for example, what's the oldest mammals that we know of? And how old do organisms live in the ocean? The answer is usually pretty surprising, because as we've delved into it, we've discovered more and more animals in the ocean that live longer and longer lives. Bowhead whales, for example. Up to about 10 or 15 years ago, it was assumed bowhead whales lived about the same life as we do, 50, 60, 70 years maybe, and that was just what everybody guessed. But something happened that changed everybody's view, and that something was this. This is a brass harpoon, and it was found in a living bowhead whale in 1993. No one has thrown a brass harpoon at a bowhead whale for over 100 years. So that whale had to have been alive in the late 1890s when it was struck and hunted and survived and carried this around for a century in a scar on its back. So this was the first view that maybe these whales are living longer than we actually thought. A very clever chemist at Scripps Oceanography named Jeffrey Botta figured out how to actually age whales based upon the decay of the lens in their eyes. Just like our eyes tend to get cataracts and things like that when we get very old, the same is true of whales. And so you can age them on the basis of those lenses. And he confirmed that, in fact, bowhead whales, especially males, regularly live to 120 years of age. 130 is not unheard of. So this makes bowhead whales the oldest known animals, the oldest known mammals, rather, with the longest lifespan. It means if you're hunting bowhead whales and you kill a 30-year-old female, you have just taken 100 years of reproductive output out of the population. And that changes how you can actually hunt a species. The same has been true in fish all around the world. Greg Caillet at Moss Landing Marine Lab, just south of here, applied a very similar kind of question to deep sea rockfish on our coast. And he found out that a rockfish about this big, a golden-eye rockfish, for example, was 80 years old to get that big. And if you're eating a golden-eye rockfish, which is marketed, by the way, as Pacific red snapper, that fish took a very long time to grow up. And it means that you can only fish that population very lightly because it takes a long time to replenish it at those growth rates. So this kind of basic information feeds directly into figuring out how we use the ocean sustainably. By knowing the age of fish, we can then get our fishing structure right. We thought of the ways that we could actually, make that into a little jingle or a rhyme or something. We couldn't do that, but we could figure out a sort of simple rule of thumb about, what sort of fish should you choose to eat, and this is what we decided, that just don't eat a fish older than your mother. And if you just didn't, a lot of these problems would be solved, and we could go on. These aren't the oldest marine creatures, though. It turns out that if you look around the ocean, there's many more older animals than fish or whales. And this is one of them. This is a soft coral. It's living about 1,000 feet deep off the coast of Hawaii. It's very dark, it's very calm, it's very steady. This animal lives in an environment that doesn't change seasonally, it doesn't change year by year, maybe century by century there's something that goes on. It grows a hair's width a year, gets 6 feet tall. In fact, the oldest of these is clocked in by my colleague Rob Dunbar at Stanford at 4,270 years old. This is the oldest animal known on the planet, older than some of the pyramids that we have built. And it just tells you that there's an entirely different way in the ocean that organisms live their lives, a different pace of life. That, again, if we don't know about it, we will use that life, we will use the oceans in the wrong way, at the wrong pace. There's all kinds of other different ways that marine organisms go about their regular day-to-day lives that surprise us a lot. And I just want to mention one aspect of this, and that's deep-sea anglerfish. Deep-sea anglerfish are surprised because they're all female. For a century, marine biologists were very puzzled by this. They were all female, every individual of every species in this group. Nobody knew where the males were, what they looked like, how they got together, nothing. Until a parasitologist, a marine biologist working at a marine lab, decided to look at the parasites that were living on the bottoms of all of these fish, and that is the male. So males have this kind of life. They're very small, they're moving around the deep, dark ocean. They find a female, they latch onto her, and they never let go. Well, before you're thinking that this is a good idea, they lose their jaws, they just melt into the female. They lose their eyes, they lose their brains. They lose their guts. They lose everything, they just end up being a dangling testis that the female uses to fertilize her eggs. They don't even get to do it alone because there's 2 or 3 of them on the female by the time she's pretty big. That's the lifestyle they have. It's very different than anyone expected, particularly since all the fish biologists at the time were all men. And it's just one of the bizarre ways that marine life works to replenish itself. The book has some, and lots of stories like this, built upon the idea that knowing something about these creatures would really help people get a feeling for them. We also tried not to do the regular narrative arc of a lot of environmental biology, which is that this is really beautiful, these animals are wonderful, they live in such incredible ways, they're in trouble, we're affecting their world, their populations are declining, and it's all our fault. So unfortunately, that arc is often true, but as a storyteller, you can't tell that story over and over and over again without exhausting your audience. So the sense of guiltless wonder in the book really is built not to avoid that, but not to dwell on that in every story. But at the end, we really do have to come back to the oceans and what the future holds for it and how we are affecting the oceans. When we do this talk together, Tony and I, he usually is speaking by this point. This is his part of our stage show. And what he points out is that the way you can talk to people about the impact that we're having on the oceans really is how that impact actually reflects back on us, because we use the oceans a lot. And if we affect them, it isn't just because, oh, it's too bad we've affected the oceans, but it actually changes how we get value out of the oceans. The story here is that this is a Filipino fisherman. He is hunting seahorses. At the time, I was working with Amanda Vincent, who's a seahorse biologist in British Columbia. We were in the Philippines, and we were being taken on trips by Filipino seahorse fishermen at night to find seahorses. We would have regulators and tanks and scuba gear and flashlights and compasses, and he would have a frayed pair of shorts and goggles that were made out of carved coconut husks and the bottles of Coke bottles, the bottoms of Coke bottles. We would both go over, us and all our gear, he and practically nothing. We would find a seahorse or two, photograph it, come back. He would spend 8 hours in the water looking for a seahorse. And if he found a seahorse, then he could dry it, sell it to the Chinese merchant in town and get 25 cents, which was enough to buy rice for his family to eat that day. And then the next night he would go out and do it again. Now, he wasn't really happy about that. He didn't want to spend all night getting one seahorse, but overfishing had driven that population down to such an extent that that's all that he had left. We see that all over the ocean. If we take too much out of it, populations drop and it hurts people. If we put pollution in, then the ocean changes its characters, it hurts people. Climate change right now is changing the way the ocean works, and it not only hurts the ocean, but it hurts us through rising sea levels, changes in where fish are, and entire changes in how the microbes of the ocean actually function. So these parts of the book are really meant to latch on to the care and interest that people have in the ocean and just say there are consequences to how we use the ocean. We know how to use the ocean correctly. That's the good thing. We know how to fish sustainably, and we know how to take care of pollution. We just don't always do it. Doing it is not just good for the ocean, but it's good and really important for us. Finally, the real message, the real way we have tried to write this is actually something that came out right at the end of writing this book. As we're writing, of course, you never write the beginning of the book first. It's the very last thing you write. And Tony and I were going back and forth with the editor, who was rejecting virtually everything at the time, and tuning the front end of the book the right way. And what Tony said really at the middle of this is like, the reason we're writing this book is because you don't care about the plot until you care about the characters. That's true of an opera, of a play, of a movie, of a novel. Why isn't it true of an environmental book? And so we built the rest of the book and structured it around the idea that you need to care about the life of the ocean to really care about the fate of the ocean. It's not hard to do because there's so many amazing things there. But what we needed to do is to present it in this kind of way. The last thing I'm going to leave you with is too short. And that's the point of it. There is still an incredible amount of life in the sea. We have access to it. We can go see it. We can experience it. And one of the most wonderful things is when our children do too. So I'm going to show you a very short video that Lauren and I, my daughter, took about 6 weeks ago on the coast of Kona in a manta ray dive. This is a dive where you sit down, there's lights, you sit down underwater, there's lights, mantas come freely into the lights to feed on the plankton that are attracted to it. And it was really one of the most amazing things I've ever done. And being able to do it with my daughter is something that you all want to be able to do too.
we will open it up to the floor, to all of you, for another 10 minutes or so of questions. So you can save up your questions, be thinking about them, and we will start out here as well. So we can give So I wanted to introduce the two students, first of all. So Laura Melroy is a second-year master's student in my lab working on sea stars on the coast here, interested in the history of population differences before and after the incredible wasting disease that's been going on here. So she's a population geneticist like Steve. And Adam Paganini is a recently finished master's student in Dr. Jonathan Stillman's lab, and he's been working on genetic effects of climate change, physiological effects of climate change as assessed through responses of intertidal crabs. His master's research just came out this week, I think, in Journal of Experimental Zoology, and there's biology, excuse me, and there's a beautiful color photo of his crabs as well there. So these two are gonna share some questions with Steve, and I'll just hang out and enjoy it.
Great. I think I'm gonna grab the microphone in case. Check. Hello. Oh, you've got one. You've got one already. Oh, you're so prepared. Why don't I even...
All right. Well, can everyone hear me? So Dr. Palumbi, like you say, your mantra for writing the book was that you don't really care about the story until you care about the characters. So what Adam and I wanted to do was kind of focus in on some of the really cool characters in your book. So like you mentioned, the anglerfish has this really neat sexual dimorphism, and we wanted to hear more about that. So if you could tell us maybe about the clownfish and the Osidax worm and how those kinds of adaptations can arise.
So the secret of that is that all the different lifestyles in the ocean are not just about living from day to day, but they also apply to mating and finding mates and reproducing the next generation. So we have a whole chapter on the strangest family lives, and they have a whole set of stories in it like that. So clownfish are pretty familiar sorts of fish, Nemo, for example. But what Disney didn't tell you about Nemo was that clownfish are sex-changing fish. They start out all male, and then sometime in their lives, they turn into females. It's not a random part of their lives. They turn into males when they first go in their first anemone. Clownfish live in anemones. And the biggest female in the anemone, the biggest fish in the anemone is a female. The next biggest is a male that fertilizes her eggs. And then the smaller ones are just males in waiting, basically. So what happens when the biggest female gets picked off is not like it goes on in the movie where Nemo is upset and he goes off to look for his mom and his dad follows him and they have adventures and meet turtles and stuff. But instead, this is the way it works. Nemo's dad grows up a little bit, turns into his mom, becomes a female. The next biggest fish, which would be Nemo, grows up and turns into his dad. And then his old dad, his new mom, and himself, being his new dad, mate and have other creatures, little babies, who knows what relationship they have to him. That's how it all works. And that's why you didn't hear about it in the Disney movie.
The sequel.
The sequel,
The sequel is coming.
you know, these days you could film that, right? So that's sort of one story like that. Other fish change sex in a different way. They start off female and they change to males. Why the difference? Well, in those fish, the males guard parts of the reef, and the bigger they are, the better they are at doing that. And so all the fish start out as females in a harem that is controlled by a larger male. And if he gets picked off, the largest female in that harem turns male, starts acting like a male within 2 or 3 days. You can imagine what that's like. And then he begins to make sperm after about a week, and the whole thing just keeps going. So that is strange, right?
So kind of a follow-up question to that would be, so I was reading the book and I was starting to think to myself, why do we even have males at all in certain parts of the sea? And yeah, and so are there any organisms that don't rely on males at all? Or?
So first, don't follow that line of thinking too strongly. Doesn't get you anywhere. But yeah, there are a couple of actually fish that don't have males at all. There's a freshwater fish that is only females. There are no males in the entire species. They're not parasites. They're not missing. These females actually get fertilized by a male of the other species, of another species. And they don't use the sperm. They just make eggs that are clonal. So their eggs hatch out into young that are exactly genetically identical to the mother. And they just go on that way. Sea slugs, pretty common around here, are neither male nor female. They're simultaneously both. They don't have males, they don't have females. They're simultaneously both. And when two individuals get together, they can both fertilize each other's eggs.
I'd like to go in and have you discuss some of the extreme adaptations that we find in the deep sea and some of the really interesting examples of adaptations such as fish that produce red light on their own.
Oh, those are really cool. So Adam's referring to a kind of fish called a stoplight loosejaw. And it's an ugly, completely disastrous-looking fish with a big, huge jaw like you expect a deep-sea fish, enormously big, long teeth. It's dark in the deep sea. There is not much food, and any food you can find, you better just eat, which is why they have these big, toothy jaws. The trouble is you would like to leak out a little light so you could find your prey. But if you do that, then your predators can see you too. And most of the light in the deep sea is made by organisms. It's bioluminescence, like firefly light, and it's a kind of blue-green. And that's what most light is. That's what most fish see in that spectrum. But the stoplight loosejaws, they are called that because they have red searchlights. Right under their eyes is a searchlight, each of them. And they beam out this red light, and no other fish can see it. But they have evolved the proteins in their eyes, the opsins, so that they can see red light. So they have these red headlights, they have night vision goggles so they can see red, and they can find their prey, but nobody can see them. So complicated, right? But that's how they work.
And my lab, Sarah's lab, studies evolution, and the driving force of evolution is selection. So one of the concepts that I found really, really fascinating was the empty barrel hypothesis. So I was wondering if you wanted to explain about that and tell us how organisms in the past differ from those today.
Tricky. So what the empty barrel hypothesis is it refers to actually a period in the past of the oceans 560 or so million years ago called the Cambrian explosion. And it's an explosion because in the fossil record there was not much, and all of a sudden there were all of these species of life that people found in those rocks from 560 million years ago. So there's an explosion of biodiversity and all kinds of things, that ways of life that exist now. Crustaceans were there, chordates were there, echinoderms were there, but a lot of stuff that died out and have names like hallucigenia. This is a scientific name of a species, hallucigenia. And they were weird forms that look like nothing we have today. So the idea was, how could that happen? All these weird things that just kind of burst on the scene. And the empty barrel hypothesis is something that Stephen Jay Gould proposed, which was that at that time in the planet, the world was not full of life like it is now. There were just new things evolving, new ways of living, and virtually anything would work. Anything goes, kind of. And that it was like the whole biological world was an empty barrel, and it got filled with just about anything. It didn't have to look good. It didn't have to work good. It just had to survive. And it would be allowed to live. Later on, when the barrel got full, then natural selection started kicking in, and there was competition, and you had to be better at it than you would before. So selection started when the barrel got full, but when the barrel was empty, then a lot of these life forms could burst on the scene and be there.
So we're running a little short on time right now, but I was hoping to get you reading this little paragraph right here in your book. If we could get you to read a passage just to wrap it up.
Sure.
That would be fantastic, and then maybe a follow-up question.
You got that?
Sure.
Turns out that by the time that we got to Star Trek movie 2, Captain Kirk needed reading glasses. So here we are, Star Trek 2. Imagine an ocean seething with microbes, its red-brown surface weedy with algae, vast dead zones and tides of neurotoxic water hamstring high-order productivity, wiping out the top half of the food chain while its base fills the world with toxic sludge. This is a sort of prediction of the future, not a good one. Waves crash on the beach, kicking up drifts of sticky green foam that cling to the sand. The clean salt air turns sickly toxic with the stench of decay. The ocean's delicate species have perished during the extremely hot days of summer or have expired in a bankruptcy of expensive skeletons.
So that's a rather ominous picture, a depressing picture of the future, and so we can all feel rather defeated in talking about climate change a lot of the time. So I want to get to know your feelings about how to stay optimistic and how you think we can improve our view of the future.
This is a tricky chapter because it really was the end, and we were really trying to be honest about the problems that are there. Because it's one thing to be glib and happy about the diversity of the sea and all the weird things that go on in it. But it is our job, I think, as scientists to say, here's the facts. They may not be pretty, but here they are, and we need to come to grips with them. We started this chapter about climate change in order to lay out the facts of how climate was affecting the planet. I am a very optimistic person generally, and I can usually think of some way out of almost any environmental problem, and we work on that in the research we do. How do organisms that live in the ocean now, how do they find a way out? How do they evolve? How do they thrive? But I found myself writing myself into a corner in every possible direction. Tony abandoned me. He said, this is your chapter, you have to write it. I'll put in the language later about the bankruptcy of skeletons, which, by the way, is a really great phrase. Talk to Jonathan Stillman about what that's about. But here's the problem, that if the planet is on its current trajectory, then we're adding more and more and more CO2 in the atmosphere. It raises the temperature, it raises the sea level, it raises the acidity of the ocean, and it increases storms. None of that is good for the ocean. If you plot that out into the future, it looks increasingly grim. So that by the end of this century, a lot of the ways the ocean is working now really probably cannot work. But we're not there yet. The ocean is still it's in trouble. There's stresses, things are problems, but it isn't broken. And so our challenge then became, how do we find a way out? How do we find a way to stop from breaking the ocean? It's not broken now. We give every sign we're going to break it by putting CO2 in the atmosphere.
We've got time.
So we have time. We went around and we found a few things that we thought were really important. One is what every 16-year-old knows about climate change, and that is stopping distance, the concept of stopping distance. You have to know that to get a driver's license. You put the brakes on in a car, it does not come to an instant halt. It takes a while. Bigger the car, faster the motion, the longer it takes. And we have a stopping distance for climate change, which is about 50 years. Even if we stopped putting CO2 in the atmosphere now, it would still take 50 years to begin to get better. So, 50 years. So that means we need to stop putting so much CO2 in the ocean by 2050 in order to roll things out by 2100, where things are starting to get better. That gives us 35 years to actually reduce emissions. And if we reduced emissions 3% a year, we would get there. We have the technology, solar panels, hydropower, wave energy is enough to do that. So that's how we ended up being optimistic kind of at the end, because there's a way out. It doesn't mean that we're doing it because us scientists can't do it, but you all can. It's the financial part of our society, the political part, just people who need to solve the emissions problem. The last part of the book is something called an epilogue. It's called a grand bargain, and the grand bargain is this: if the financial, political, and social part of our society will commit to figuring out the climate change problem in the next 35 years, so that in 100 years things are getting better, then the scientists, the conservation biologists will commit. Our part of the bargain is to save as much of the planet as possible. And that's the bargain. That's the way out. I think it can work, but it's not going to be particularly simple, and we all have to kind of join together to do it.
Well, on that amazing note, we should just go out and do that now. I have to say, I can't think of a more eloquent way of putting it. But I'm sure that we could use some details. So that's up to you guys in the audience now. There are 2 mics. Ian Slaughter and Tony Ignafo will be circulating around. If you raise your hand, they will bring you a microphone. Maybe it's this microphone, actually. Oh, okay. So if we see some hands raised, I'm sure Steve, there we go. Oh, here's okay. Do you want to give her a mic right there?
Hi. My question is, could you talk a little bit about what it means to sustainably fish on the West Coast?
So the question is, what does it mean to sustainably fish on the West Coast? It's the same as sustainably fishing anywhere. It means that you balance the fishing effort and the fishing success around what it is that population of fish can produce. And when it's in balance, then 2 things happen. Actually, the yield is pretty high compared to often what it is now, and it's forever. It's not a given stable number. It varies with the environment and with the year, but it's a balance. It's a balancing act between what the fishery can produce and then what we're taking out of it.
And do we have that information now to be able to promulgate that to fishermen?
So the question is, do we have that information now to be able to transmit that to fishermen? And for some species, we do. We do have that. We know pretty well for those rockfish, for example, that I was talking about, what the balance is and how we can fish them. Rockfish, those rockfish were put on the threatened species list along the whole West Coast. They were taken off a couple of weeks ago. There's still concern, but there has been a lot of improvement in that. One of the troubles is in a zone of contention, really, is that you're never really sure. And you're never really exactly sure what the fish population is doing, how it's working, and so that uncertainty leads to conflict about what the recommendation is. Some folks want to look at that uncertainty and say, you can't prove to me that I shouldn't take this number. Other people look at it and say, really, we should take that number in order to be sure. And that's where I think the conversation gets into conflict. One of the things I learned from writing the previous book, which is about Monterey Bay and its recovery from harm, is that fishing now is totally different than it was 100 or 200 years ago, because then it was just take, 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 take. Here, all the fishermen that I know there's a limit. The difference is what the limit is, and that's where we do have to work at it.
The starfish, I read that they're dying. Is that in Hawaii? In Hawaii too?
The question is, are the starfish dying in Hawaii too? And the answer is no. That the starfish wasting disease that Sarah mentioned a few minutes ago seems to be restricted to the west coast of the United States. Ironically, there's an outbreak of crown-of-thorn starfish in American Samoa right now. Those starfish are eating the coral reef, and American Samoa is trying to kill those starfish. Just irony.
Could you explain why the cod is not recovering in the Atlantic in the Northeast?
No. No, I'm sorry, the silly answer, but it's one of those strange mysteries because there were so many cod and we fished them so long, centuries, and the numbers went down and down and down and down, and then we really got an idea that that was too much and pretty much stopped. Cod fishing was really reduced, but the numbers didn't come back. And there's a couple of hypotheses. Some people have said, well, actually, instead of one big cod population and that you've taken 99% of, there were actually 100 cod populations, and you've wiped out 99 of those. They're extinct, and they're not coming back. And so that's one of the explanations that people have offered, that our view of what the population is was not the fish's view, and that we may have wiped out whole populations that have no chance of coming back.
I have a question. I understand that global warming of the atmosphere is made up of two parts. One is the CO2s, and the other is the feedback that magnifies that effect. How about in the ocean? Is it only the CO2, or are there feedback mechanisms that magnify it or offset it?
The warming of the ocean is in equilibrium with the warming of the rest of the planet. So the same feedback mechanisms that work on warming the atmosphere also warm the ocean as well. There are a couple of other things, though, that happen in the ocean that affect that. For example, when the ice sheets melt, when the glaciers melt, then the reflection of all that ice stops. And a lot of the light that's hitting the planet up there is reflected back into space, so it's a little bit insulating. Without that, then the ocean warms a little bit. Another thing that's happening is that most of the shipping that goes on in the world is, it's very cheap. Why is it so cheap to ship stuff around the world? Because those ships use the dregs of our oil production, the cheapest, dirtiest bits of petroleum, and there's a lot of soot there. And so as ships move around the planet, they are dumping soot out into the air. It's falling on places like the Arctic and the Antarctic, making the bright white snow more heat absorption, and then making the warming even more. So those kind of things work in the atmosphere too. The thing that's really unique about the ocean and its effect from climate change is that CO2 in the atmosphere dissolves back in the ocean. That's normal. But it turns into carbonic acid when it gets there. And that carbonic acid makes the ocean more acidified, drops the pH, and that makes it harder for ocean organisms to make their shells. So Jonathan Stillman's group and some others here at the RTC have been really pioneers in studying that process. Your paper that came out this week is based upon those kinds of data. And that's a unique feature of climate change that only occurs in the ocean. And if we stop the warming trend by somehow blasting Mylar into space, don't do that, by the way. Suppose we did. Suppose we decided to do that. It wouldn't solve ocean acidification as a climate change effect.
I've been told this is the last question. I'm over here.
Can you give us a prognosis on the future health of a very big and smart marine mammal, namely the orca?
Right. So the question is about the prognosis for a really big and smart marine mammal, the orca. And the prognosis for the orca, there's good news and bad news. The bad news is that orcas are at the very top of the food chain, And a lot of what we put into the ocean then is accumulated in them in terms of pollutions. Dioxins, PCBs, heavy metals, all get accumulated in top predators like the orcas. They have been lucky that they're feeding in northern and southern latitudes where those problems are not so big, but now things are happening to cause a pollution problem there. The good side is actually the movie Blackfish. It was an emotional shock to many people that these captive orcas had so much behavior, and that they should think of them as being different than just some performers in a zoo. So that movie, although there's lots of controversy about it, really changed the game about how people thought about the wildlife that's in the ocean, that it deserved to be wild, and that some of the things we were trying to do with that wildlife had consequences that we could really empathize with. So just in a nutshell, that those are two things that are happening. Orcas are is a global species. They eat lots of different things. They're clever. They're adaptive. So in terms of their future in climate change, I think they actually have a pretty good thing going. But there are threats, particularly for pollution.
Great. I think we're there. Thank you so much to the panel, to all of you in the audience, and to Steve.
Thank you all so much for coming. You have all been amazing marine biologists and asked very interesting questions, and we hope to see you again in the future. We have our spring public forum scheduled for April 15th, so please mark your calendars, and if you're interested in receiving notification about those events, you can sign up on our webpage if you haven't already. And I just want to thank Richard and Barbara Rosenberg again for allowing us to put these forums on, and it's really a pleasure to have you all here. Thank you.